Thank you, Rolf. Actually, I will tell a story not only from Diagnostica. There will be examples from Diagnostica, but also from some of my previous employments in the diagnostic industry. Actually, there will also be examples of different technologies, because at Diagnostica we are, as we also heard from previous presenters, very interested in blood sampling. But on the other hand, some of my previous works actually were in software diagnostics, and before that in slide-based analysis, as we also heard about today. Not genetic slides, though, but slides for immune antibody-based reactions. So I'm also here with another purpose. I have to say that straight up here at the first part of my presentation, that I really hope to see some of you again later. Some of you who have good ideas for biomarkers or other types of products, particularly within the interest sphere of Diagnostica, but even if slightly outside that area, and I will come back to what area I'm talking about, but even slightly outside this area, we are always interested in hearing about new inventions and good ideas for diagnostic products. And if we cannot help you, we are certainly having other companies in either within the same ownership or even outside our ownership who could be your next partner. So I'm trying to give you some examples of what we expect from academic partners, what we hope to get from academic partners, and on the other hand, explain to you what we can offer and what we think are important parameters to have a successful products coming out of a biomarker. So I'll give you a very brief introduction to Diagnostica. I don't want to spend too much time on that. We only have 25 minutes. But I mainly want to talk about what it takes for a biomarker to become a commercial success. And we already heard about this. The unmet need is really probably the most driving force. We also have to think about competitors. That has to do with how much commercial success you get in the end. Actually not so much about the technology. And then there's how do we actually try to project or calculate the financial upside. Even many years, as you heard, it takes many years from the idea is generated to the first prototypes and so on before you actually get to a product. But already very early we have to try to find out, is this going to be a commercially viable project? Should we do the project at all, seen from an economical point of view? So I think there's one knowing about marketing and sales, but probably the rest of you have not so much, I guess, please correct me if I'm wrong, not so much insight into how you calculate projects, how do you calculate the economics of the projects, and how do you compare two different projects to find out which is the most financially reasonable project. And of course, that is not a very exact business to do that. There are many factors which have to be included. Some of them are not easy to quantify. For instance, project risks. So Diagnostica is mainly operating within the field of autoimmune diseases, and rheumatoid arthritis being probably the most important disease for us, but also vasculitis, and actually to a certain extent also type 1 diabetes. We are based on small companies existing already in the late 90s and late 80s and early 90s who specialized in small, peculiar, special assays for certain autoimmune diseases, but they combined their forces and became thereby Diagnostica, which is still a leading company within this special disease area. So we hope to be a partner of choice, not only for universities, but actually also for clinicians at hospitals and even for other industries within our field. Beyond actually working on our preferred field, we also have capacities at our company, which is just 5 kilometers away from here, or 3 kilometers away from here. We have capacity for manufacturing, particularly assays which are similar to our own, but in other disease areas, for instance, infectious diseases or allergy. And we actually produce for several other companies regulatory approved and clinically approved diagnostics. So they are labeled in the other company's name and sold at different markets. But since we have this well-developed manufacturing system, we are very often chosen for small companies who don't want to set up or do not have the capacity to set up their own facilities. They can sort of rent our facilities and competences to produce. And finally, and probably most important for the success of this company, we are very special in having a clinical service laboratory. So many autoimmune disease diagnostic tests are actually made in our company at a certain part of our company called VisLab. It's a clinical service laboratory. They earn money to do the testing for other hospitals here in Sweden or even Northern Europe and Scandinavia, submitting samples, very frequently samples for odd kind of autoimmune diseases, small diseases where you have to have a high level of specialty knowledge to do the testing. Not so much for routine, high throughput, hundreds of tests per day, but more the very specialized tests. And we are therefore also able to collect those samples. We are allowed to store samples, and we have more than 80,000 samples in our bank coming from autoimmune diseases, particularly here in Sweden, over the last 20 years. And since the recording system of health status and so on is very superior here in Sweden, we actually have a lot of information together with those samples. So as you have heard today, it's really, really important to have access to clinical samples. And it's also very important when you develop a test that the early stages of it, you have a prototype and the next prototype and the next prototype. You can test them upon sort of clinical relevant situations. You can put them into your own lab, run them aside on the tests that were actually required or acquired from your customers. And we can keep the results, and we are allowed to do that. We don't share those results, however, with the doctors, because this is not yet a test which is accepted for clinical use, but we get the knowledge during development, and therefore also we are a preferred partner for many companies which are much larger than us, who do not have this easy access to samples. So if you're setting up your own company, you should actually think about maybe doing also aside a clinical service laboratory. It's not an easy thing, but it's really valuable. And probably because of all this, we actually this year got the award from Frost & Sullivan, who judged that being a companion either for other industries or for academia, we are very well suited for doing that in the autoimmune disease area. So I'm not going into details about this final slide about Eurodiagnostic. I just want to mention that these 4 companies were the early merger. There have been several companies added during time over the last 20 years. Again, you can see that VisLab probably is the most important part of our company for what concerns the development of new assays. And I want to say also that here the most recent years we have moved from being very focused on diagnostics to also have tests for prognostics, but maybe even more for prediction and monitoring disease treatment. So when your patients are in treatment with particular new biologicals, how can you assure that the patients still respond well to the drug, and how can you switch them to another drug if, for instance, they generate autoimmunity or immunity, to this drug. So I'll give a few examples. The first one is from Eurodiagnostica. It's a diagnostic test, and we'll talk about how it is to be first in class versus having the timing right according to the end user acceptance. I'll also give another example from my past, actually with combining software and reagents for diagnostics, thereby actually serving a complete solution. I want to give you some general considerations about why it is important to patent, but why it is not enough to patent, and that you also have to consider freedom to operate. And finally, I'll give you just a very brief idea about how we try to calculate whether a project is superior to another project from an economical point of view. So this first example actually is based on frontline research done by scientists headed by Professor Van Wanrooij in Nijmegen University in the late 80s and early 90s. They really got a good understanding of the autoantibody response that you have in rheumatoid arthritis. And late in the 90s, they published a very important publication showing that rheumatoid arthritis is characterized by early generation of antibodies which react with citrullinated proteins. And citrullinated proteins are proteins where one or more arginines are converted into acetylene in particular proteins, thereby becoming a strong immunogen for the raising of autoantibodies. And it has a lot to do with joint problems in rheumatoid arthritis. So you can see here the publication, what it has generated, more than 200 publications every year if you search PubMed on Situ-related peptide and rheumatoid arthritis, coming out of basically nothing before year 2000. Fortunately, and because they collaborated with Diagnostica, they were urged to make a patent application before this publication came out. And they actually started by looking at Situ-related peptides, but they found out with our assistance that it was very nice to have the antigens presented not just as a single linear, small linear peptide, but actually as a cyclic peptide, thereby exposing even more the citrulline moiety, and therefore by actually measuring antibodies towards such amino acids. And I have to maybe emphasize that the anti-CCP test is actually exactly that, an antibody test. So the biomarker is not the CCP. CCP is the antigen for which we measure the biomarker, which is actually antibodies. So it's not a single molecule, it's a whole panel of high and low affinity antibodies which are directed towards CCP. So it's not as straightforward as if you have a test where you measure one gene or one antigen in a sandwich ELISA construction, for instance. But here you have to measure a wide panel of high or low affinity antibodies, early immune responses where you only have a few clones producing antibodies if you want to have a test which responds early. Okay, so soon after the publication, they also had a license contract with Diagnostica, which allows us to develop a commercial assay based on the CCP antigen. And the first test was launched in 2000. Actually, very few customers, only some convinced persons. You can see there were no publications at that time, so we didn't really have good references. Only those which were very early adopters dared to bring this test into their clinical settings. So in the meantime, the scientists at Radboud actually continued their work with cyclic citrulinated peptides, and out of 12 million peptides screened, they found a certain class which was really, really good as an antigen, high specificity, and actually picking up more than 70% of the rheumatoid arthritis patients. But most importantly, almost no of the reactions were false positives. So you could really believe you are a patient with rheumatoid arthritis if you have a positive result. So a new assay was launched in 2002, and at that time, also because the test was better, it became much easier, and it became also a commercial success. And some of the companies, that's probably one of the best measures if you're a small company, and some of the big companies like Roche, Siemens, and so on come to you and ask, can we have this test as well? Sure, you have to pay, of course, but it's much better for us as a small company to have it on their instruments. They have maybe 1,000 or 5,000 instruments on different laboratories around the world, and that's a very fast way to get access to large customers in hospitals and reference laboratories. So we have generated substantial income from this test, and we have shared that, of course, with the original inventors through royalty payments, both from the sales that we have directly by selling an ELISA, but also through these business-to-business sales. So, and what we hope for, but that takes a long time, and that's not still not yet achieved, is that we want to push out the old biomarker, rheumatoid factor, very, very popular, super name. Actually, we should have had that name. But the rheumatoid factor is really still the easy one to cross off. If you sit in a laboratory, you come to a patient, do you want to have a test? Sure, I want a test for rheumatoid factor if I think the patient has rheumatoid arthritis, even though it's a very poor marker. It's not very good. And it has been around for more than 70 years. It's adding, but it's not a valuable single marker. So at this stage, we have more than 15 million tests worldwide being made for CCP. Quite well done, actually, within 10, 15 years. But there are still 50, 60 million rheumatoid factor tests being made. And it is cheaper, and it is more automated and so on. So I, of course, accept and understand that this is still important. But I think what we are really aiming for is taking over the entire market from rheumatoid factor.
Why don't you call it rheumatoid gold or something?
Yeah, yeah.
CCP.
I can tell you, the name is crucial, actually. We have now a competitor who has invented why not CCP3? It's a poorer test than CCP2, but it's still CCP3, and everybody expects the one. So yeah, it's tough with names. Okay, so this was a commercial success. Have that in mind when I now tell you about this story, because we were so proud of the ELISA, we were proud of the antigen we sold to other companies, that we were looking for other areas where our product also could be used. And then we had this assay developed, a home test. You can actually do it at home, it's easy. And you can definitely go to your doctor's office and have this test made. It has a lot of technology benefits. We spent a long time, not me, I was not there at that time, but it's a really good product. The test can be done in 10 minutes like a pregnancy test, though you have to use whole blood or plasma or serum. It's simple, it doesn't need any tools. It's accurate, it's as specific and sensitive as the ELISA, and it's easy to store, and actually also patented, so we didn't have any competitors. However, even though we were first, and still only, except for some Chinese competitors who try to move in, first is not always the same as right timing. The market was and still is not really ready for this product, and we still wonder why, actually, and we try to work with this truth, and we have to convince smaller niches of the market that this is interesting. For instance, now we have in Germany a huge lorry going from big company to big company and doing all kind of staff testing for all kind of diseases, and they now have this CC point box, and if you are having some rheumatoid arthritis-like symptoms, you may be tested by this test, and you will have your result in 10 minutes. But it's of course a very cumbersome way to get your product on the market, and of course also it has to do with the price of the testing. So this test costs about 3 times more than what a laboratory pays to do an ELISA. So, but we cannot make it much cheaper actually. It's a quite expensive format, and
Yes.
that maybe, yeah.
What is the most common argument someone is using why not using this? Is it price or
Yeah.
what?
I think it's price, but also you have to remember, again, we have to understand what do you actually do if you get a positive result. I mean, if
Yeah.
you're pregnant, you
Yeah.
may know what you want to do. But if you get a positive result,
Yeah, that's the.
And you have no symptoms.
and no symptoms, it's at least it's easy. Yeah. Probably if you buy such a test, you do have some expectation, or at least your mother had rheumatoid arthritis at your age back then. Or yeah.
So what's the price?
Around 15 euro.
15 euros.
So
And you can buy it in Apotheke.
yes, you can. Large apotheques, not all.
We have one buyer here. So,
Okay.
what is the predictive power of this test?
Yes, as I mentioned, the CCP is not picking up all rheumatoid arthritis patients, not even as ELISA or high sensitivity assays. It's about 70% of the RA patients who are positive. They may be positive at a very early stage, so pre-symptomatically, and it's a very good test because if you are positive, you will have rheumatoid arthritis. But 70% only, and probably it's because rheumatoid arthritis is not just a single autoimmune reaction.
But I think we need then to ask about the specificity. That's for me even more important from an ethical point of view. So how many non-diseased will you pick up by the assay?
None.
None. Okay,
None.
so that's
None. But I think that's not a problem for us since we have this very specific result. But it is of course the sensitivity, because if you get a negative result, are you then thinking for the next 10 years, I'm not having rheumatoid arthritis, even though you start, having all the symptoms. I made the tests, and I know I was negative, and I can make the test again, and I'm still negative. So you have to explain that a negative is not a guarantee. Okay, I'll tell a completely different story now. It's about screening, first of all, so it's a biomarker for screening. It was made by scientists who for more than 30 years had worked very hard to understand testicular cancer, headed by Professor Skakkerbæk at Rigshospitalet in Copenhagen. And, can I go back? Yes. So this group had a lot of understanding of the basic nature of the disease, and I think that was crucial to this project. But also they saw that there were a certain part of the solution which was required that they could not add themselves, and therefore I want to highlight this collaboration which was done in order to succeed with this project. So first, very briefly, what is it about testicular cancer? It's actually a rather frequent, but also relatively kind type of cancer. It hits 0.5 to 1% of the male population. More and more, the frequency is, or the prevalence is going up enormously, even threefold within the last 50 years, and has probably a lot to do with environment during mother's pregnancy with male children. Come back to that if you have any questions. Also related to that, the risk is 3 to 4 times higher to get testicular cancer if you are a man experiencing infertility problems for more than 1 year, which means that you're trying to get a baby for more than 1 year and you don't succeed. So either being infertile or subfertile. So there is a preceding stage to the testicular cancer called carcinoma in situ, which is what actually this group at Rigshospitalet studied very carefully. And actually, we now know that it's a precursor for testicular cancer, and if you do not treat at this stage, you will definitely develop testicular cancer. That takes some years, but then you will have the classical symptoms of testicular cancer, and you can substantiate your diagnosis by ultrasound examination and biopsy. 50% of those who have testicular cancer still find it too late, so the disease is disseminating, and on the other hand, treatment is fairly good still in this situation. So it's an expensive treatment. You may have lifelong hormonal treatment, and you risk all kind of secondary issues. But just to say that very few actually die from testicular cancer nowadays. If we were able to detect carcinoma in situ, it would be much cheaper to treat, and the risk of other secondary events would be substantially reduced. Oh, sorry. So what I want to say is that the finding that this group made was that according to immunohistochemistry, there are cells in the testicular tissue of those few patients known to have carcinoma in situ before they actually get testicular cancer, who have this transcription factor. Fetal transcription factor shouldn't be there at an adult stage, but it is expressed in the nuclei of carcinoma in situ cells in testicular tissue. Again, this time on their own, they were clever enough to actually patent a whole panel of different potential biomarkers of testicular cancer before they published this data. And very importantly, they also had a publication 1 year later which showed that some of these cells are actually exfoliated into the semen. So you can find the cells, very few of them, but still some of the cells in a semen sample. So unfortunately, we are not working on blood samples. It's definitely more inconvenient to study semen. On the other hand, you have to remember that the main risk group is men with infertility, so they usually will have a test for their sperm quality anyway, and we just need a small sample. So what we're doing now, instead of doing immunohistochemistry and avoiding actually this invasive procedure of having a biopsy, testicles, you can see some of you looking. That's unpleasant, yes. In immunocytochemical tests, so you can have an early identification of carcinoma in situ. You can do it by a non-invasive method. Unfortunately, the method is very cumbersome because you do a cytospin, you do the staining, and then you have to do high-resolution microscopy of a relatively high volume of semen and semen cells, sperm cells, and it takes about 20 minutes per slide, and even a skilled and thorough technician will not be able to do much more than 20 or 25 slides per day. It's also a boring job. Only 1 out of 150 slides. I mean, if you're in the laboratory, you want some positives. Probably if you are the patient, it's opposite. But only 1 out of 150 slides are positive, and even if you look in the high-risk groups. And even then, only 2 to 10 cells per slide. So it's almost like finding a needle in the haystack, or probably more like looking for Waldo in a huge park of a lot of people. Yeah. So that was when the scientists came to the company I worked for previously, Visupharm, an image analysis company involved in handling and quantifying images from microscopes. And we were quite fortunate because at the same time, a big step in instrumentation, moved us from microscopes, which had been used for 300 years, much better later than sooner or before, but still very cumbersome, you know, sitting over a microscope, even in a dark room and so on. Now you can do whole slide scanning. You can have a huge digital image, and you can do your analysis on the digital format. So that was just at that time also. And at Visiopharm, we told the staff, the scientists at Rigshospitalet that looking for this cell type in a semen sample where there's a small nucleus with something red in it, it's really, really hard for even a good image analysis program. You need something more to sort of get your cells clear into the background, compared to the background. So they actually developed a double staining test, so the cytoplasm is blue and the nucleus is red. And as you notice here also, even these are, of course, picked images, so I took some nice ones. These cells are not looking so nice as you may see from cultures and so on, because these are cells which are released from the testicles into the into the semen sample, and they should not be there. And it's a crude environment. It's not a place for cells to keep their morphology, so there are enzymes and all kind of stuff which should degrade those cells. So they don't look very pretty, and not after cytospain either. Okay, so this is how the procedure goes. Semen is collected, probably taken from a test anyway due to the infertility problem. They are spun down on the glass slide, and they're double stained even automatically by instruments. And also then the whole slide scanning is done almost automatically. So you end up with a huge image, several gigabytes, which needs to be analyzed at high resolution. See, so I'll just show you here how it looks. We zoom in slowly into the preparation. You can just see the sample was there. And when you get closer, there starts to come some morphology. But you have to think about looking in microscope day in and day out and looking for this type of single cell among so many sperm cells and blood cells and so on. It's hard work. But that's what we want to find, anyway. And this is the result, which, as it comes out from the computer does not try to make the final conclusions. It says, for instance, you can say, I want to see the 15 most likely positive cells, and you will find, I don't know if it's 15 or 20 or whatever, and you will even grade them. So a few of them are labeled P and red as positive, B for borderline, or probably negative, and this is how they look when then presented to the final reading. So you spend all your time, instead of doing microscopy and screening, you spend all your time to judge cells which were picked up by the computer program. So we had to confirm, of course, that this way of doing it is sensible, and this is the first and until now only published study on asymptomatic men with subfertility problems or infertility problems. 765 men, 1,165 semen samples, fortunately only 11 positive samples, and coming from 5 men. And 3 of those 5 men have now been confirmed by biopsy and treated for the early testicular cancer. 1 of 5 could not be confirmed, and we, of course, still monitoring this person. And 1 of 5 did not want a biopsy, so maybe he will come back someday, I don't know. And also we tried to test, and that's a small study, and it's not easy because we actually tested how well does a test then works if you have a true positive. So we had to go to the testicular cancer patients. We could not really find the carcinoma in situ patients easily, at least. So 14 out of 22 men in a single semen sample from each of them, 64% were positive for these cells. So now, actually, the business plan is with Kirsten, at least with your company, and because it's a public innovation, it's made by Rigshospitalet. It has a private partner, Visiopharm, but what we really want to do to make this test come out is going to one of the large scanner producers or going to those companies who offer the IT solutions to hospitals. So this is not for a small company like Visiopharm and neither for an academic institution like Rigshospitalet. So just to sum up, be first in development, that's always good, but not necessarily an absolute for success. Provide complete solutions. So if you have a biomarker, think about your friends in software, instrumentation, or try to make companionships with those who can ease the process, can add to it. Also, for instance, what is very often forgotten is even if you have a relatively simple test, not a multiplexing test as we heard about earlier today, but just a simple test, what are actually the conclusion can be drawn if you have this level of result and this level of result. Try to make an algorithm so it becomes easier for the doctor to make the final diagnosis. So that can just be a table. If this, then that. If that, then this. And then you have to schedule your product launch. Either you're lucky, there are established end users who have needs, or you have to create needs. I think we can look to marketing there to try to create the needs if it's difficult to see the need today. And actually you have to look ahead in time because there will be years until your product is on the market. And very often these are also driven by regulatory demands, so having the right applications and so on. So I have two more subjects. Can I, how much time do I have?
Yeah.
Can I do that? Okay. So I will give a general introduction to patents. I don't know how many have any of you tried to make a patent application? No. Okay.
In this room, but
No.
a few of the scientists here have.
All right. But then I'm probably at the right level, I hope. And I'm not an expert. I'm not a lawyer at all. But I have been in their hands, and it's a tough world to get into, actually. So there are three things which have to be there if you want to patent your invention. It must be novel. It must have an inventive step, and it has to be applicable to industry. Novel is the most easy part, actually, because it's an absolute measure. Did your invention come out in this patent filing before anything else? You should say it's an easy yes or no. Sometimes there's a lot of fighting about that, but it's at least something you can measure. I'll give you an example. It has nothing to do with biomarkers. It's a very classical example that you think something is new. Actually, this guy, Krøyer in Denmark, made a lot of inventions. He's very good at making inventions and had done inventions over many years and all kind of different inventions. And he made also this invention that if you want to have a sunken ship coming up, you can pump balls, small air-filled balls, into the trunk of the ship, and it will come up eventually if you have given enough of this air to actually pull up the heavy ship. But the patent examiner looking at this patent, not the first one, but the one down in Netherlands, he was a reader of classical comics, and actually he found this Carl Barks story 15 years earlier. This was already invented by Donald Duck, or at least somebody around him, and using ping pong balls. So, and Krøyer has for all his life, I think he's dead now, but he has for all his life said, I don't think I read this, but of course he may have read it and just forgotten and got inspired. And it's a very good invention. It is being used to take up sunken ships, but the patent is not there. So for novelty, there are do's and don'ts which you should have in mind. Some of them are obvious. What you should do is, and we already heard about that too, check what is actually the present public knowledge. You may think you know your field very well, but you also have to look strange places, and maybe you don't really have time for that. So there are patent agencies who are really good at making searches as long as you help them to use the right words and so on. And they may not look in Donald Duck magazine, so I'm not sure.
Well, here, just to add, you should talk to Annie first. Before.
Okay.
And
Okay.
Annie will do all this.
All right, great.
You know that already.
So you may also find prior art, as it is called, and you may then adapt your invention so you don't get in conflict with prior art. Or alternatively, you may accept that you are under prior art, but you still add even more invention into it, and then you can have a situation where you do not have complete freedom, but because you have to pay a license to this invention which is above yours, but at least you have the ability to protect your invention because there is even more in it. You can also schedule your submission of article. You already saw a couple of examples. The article actually in one case came out just a few months after the patent was filed. So you can submit your manuscript. Just tell the publisher, don't put it on the internet and don't publish it before I give you green light. Do not, I've written many articles where I love to have this in my conclusions. future investigation. This is a way of, I didn't do it, but I am going to do it, and probably you should not, the other guys should not do it before I do it, because I already suggested I will do this. And that's okay, but don't put your invention and ideas in that future investigation will show sentence when you write your paper. Be careful when you present your invention at conference and public meeting. You can come around the subject perhaps, but don't mention the invention. Now we have many more risks because of the internet. You may indicate in Facebook, which has a very short duration. Maybe it's not easy to find even a few days later, but it has been published, and if somebody took a print or somehow conserved it, it is still a publication. If you make grant applications, most of it is confidential, but some will be used for short summaries or be repeated in public. It's really dangerous. And if your biomarker is going into a clinical study, even though it's an immature biomarker, indicated in the study plan, then it's also becoming public. At least you know more about that. I'm not quite sure how much of the clinical studies are actually published, but you can find some. Yeah, you know that, Kirsten.
Yeah, the last 3 incidents I have had where people have destroyed their own patent has been in clinical trials where because you need to make it public on clinicaltrial.gov
yes.
in order to later get it published, they actually destroyed their own invention. So be very careful when you publish your clinical trial.
Okay. These two, I will not use so much time to just try to describe what is inventive step. It's a surprising solution to a technical problem, and so surprising is a very difficult word to measure, and that's why it's provisional, and that's where there's a lot of lawsuits. I said that before you said it, and so on. Or I indicated this together with that, then you could make the solution. So it's usually something with combining information from here and there and saying it's not obvious or it is obvious to combine this information. Industrially applicable, that's probably the most surprising part of being patentable. Not so much that an invention made or used in any industry or agriculture is actually okay, but what it is not. You cannot patent a method of surgery, therapy, or diagnosis. So the method on how you make the diagnosis on the patient as a doctor cannot be patented. That's in order, at least the argument has been, everybody should be available, the best treatment and therapy and diagnosis should be available to everyone. But it's okay to patent the products, the instruments you use for surgery, or the diagnostic kits, or the software that you use for diagnosis. So there's still a lot of room to do patents. Don't let this stop you. You should not just tell how the doctor make the conclusion based on your test, for instance, in the patent. So probably the most important thing about patents is that they only give you a limited right. Even if you have a granted patent, it's geographically restricted. It's only in the regions where you pay to have the patent that you are protected, and it exclude others from making, using, or selling the patented product. So even for developing similar products. But the granted patent is not a right for you to make, use, or sell the patented product. That requires freedom to operate. So if somebody has a patent above yours, you actually have to have a license to get into the commercial market. So very useful example is Organon. It's an old example though, but Organon patented general principles of ELISA back in 1970. I think they actually cheated some Swedish scientists who were there before that. They were almost similar in time, but the Dutch patented, the Swedish did not. They thought it should be an open, useful method for everyone, but Organon didn't think that. So they had for 20 years all patent rights to ELISA. That didn't stop other companies from making their specific ELISA products, even patenting them, and they could also market them, but they had to pay a certain percentage for every sale they had to Organon. And fortunately, I have to say, Organon was very fair. They had a reasonable royalty scheme, which did not stop many, I think, from producing their own good products. Then came a more tough guy into the scheme, and that was Hybrid-Tech, because that was not a big invention, in my opinion. They just took the monoclonal antibodies, which were there for 20 years, only for 5 years at that time, for 5 years, and said, if you make an ELISA based on 2 monoclonal antibodies to measure an antigen, so-called sandwich ELISA, then it is a special part of the Organon invention, which can have a separate patent So hybrid-tech has to pay to Organon, but hybrid-tech also protects everyone who wants to have a central ELISA for a specific marker. So, and that was much more fierce type of royalties that had to be paid to hybrid-tech, and in the end there was a lot of lawsuits, but actually hybrid-tech won that. So in 90, Organon disappeared because you only have a patent for 20 years, and in 99, finally hybrid-tech disappeared as well. So I want to spend just 2 slides on how do we...
There's one thing for this. What is very important, you also said the use of. That does not cover research. It's a commercial use.
Yes,
So you will never be restricted by any patent in your research.
that's a very good point. Yeah. Yes. Okay, so I just want to spend a few slides on how do we then calculate the value of a project which should become a biochemical, a biomarker for diagnostics. And what we try to particularly look at is what are the investments we have to make, not those we have had made, but only the future ones. What are the risks associated with what we are doing in the future, and what are the profit we would expect to get from having a product on the market. So we had to budget investments, and we had to estimate risks, and we had to find a way to mitigate these risks. And there are a lot of parties involved in our own company, but also with our partners who need money to go through with this development. Of course in R&D, but also, as we heard before, key opinion leaders and advisors from outside have to tell us what should this product look like. And they may not be so inventive as us, and they may be conservative and say, oh no, you will never succeed with that. But at least you get their opinion, and you try your ideas with those experts. And probably also later on they will help you to do the clinical studies if they are good advisors, and they will help you to market your products by being the first who can make the scientific publication and document that your assay work. You have to spend money on patenting. It's not so expensive to start with, but it becomes more expensive later on. And you have to pay licenses to companies who have superior rights. One thing which is very often not considered enough is the problems of making a production transfer. So you have an assay, it works nicely in your R&D lab, but suddenly you have to produce 3,000 ELISA plates and put them in boxes and ship them, and they still have to work after that. And that may not be so easy. Then clinical studies, we've talked a lot about that. I don't want to talk more about it. And regulatory approval, I haven't talked about that, but I don't have time to talk about it. So we can come back maybe to that later on. And then, of course, there are costs associated with launch and marketing. So you have a cost profile over maybe over 10 years, of course, very uncertain, but at least your experience can tell you that you are not too far off with these projections. And then, of course, you also have to find out what are the sales. 2 parameters are very important. What is the incidence and prevalence of the disease? So how many are there who can be tested, and how often should they be tested? And also try to estimate the competitor situation, not today, but when your product gets to the market. There are sales through direct channels, where you have all the profit, through distributors, where you will only get part of it. Business to business, also again, you get only part of it. On the other hand, you may get money early on where you need them for your investigations. They may pay you quite early, those industries that you work with. And finally, not so economically important, but that creates data. You may have services to provide. And then you have an income scheme, no money for the first years, except if you have grants or some of these upfront payments, but otherwise income. Actually, it's not just the sales. You have also to think about what will be the cost of the goods. I mentioned this CC point, quite expensive to produce, and you have to subtract that to really understand what is your profit. And this is the last thing I want to just mention. You have then to calculate the net present value of your project. So you take your sales minus the cost of goods, minus the cost of doing the development, and then you have to know that money spent today are more expensive than money spent later on. Money earned today are more valuable than money earned later on. So you have a discounting factor, a basis rate, perhaps 4%, something like inflation, a little more, and then a risk premium, which is artificial, but you can say if it's a low risk project, you may only have a 2% discounting factor. You add those two and you look at your expenses and income, and you have the annual result in blue, and you have the accumulated, and you can add them because now you have corrected for some coming late and some coming soon. You can add them, and you can see that in 2018-19 you're going from negative to positive. So that's your return, where you have your return of investment. And then you can actually look 10 years ahead. Your value of this project is 1.4 million euro. If it's a high-risk project, you probably put another higher discount rate on for the risk part, and you can see that it takes longer time to reach the return of investment, and that your project is actually in a 10-year period only less than 1 million euro. So if these were two projects, you would definitely go for this low-risk project, but it may not be the only parameters, of course. So I think I will stop here and just hope that it hasn't discouraged you from working with industry. And then please come back if you have any good ideas. We are always open to also very informal meeting. You can have a confidentiality agreement with us, so you can also tell us about ideas, and we will not misuse them if you go to another place. Okay.
Thank you.